Hello students, this is the third part of industrial microbiology and I am going to discuss the enzyme technology here. Basically in this part I am going to describe the what is enzyme, and after that how the enzymes are produced in the industry and what are the uses of those enzymes in the industry, and how we can use those enzymes to produce products of a particular substrate in the biotechnological industries. So let's start. Now, first question arises: what is an enzyme? The enzyme is a biological catalyst that promotes and speeds up the chemical reaction without itself being altered in the process, and they fulfill their role by binding specific substrate and at their active sites. So basically, the functions of enzyme are it can degrade or build some products by acting on the substrates, and these two are categorized as the metabolism. So when the enzyme builds or synthesis something, that is known as anabolism, and when it degrades some substrate and the product is formed, that is known as the catabolism. And apart from these, the enzyme can also modify the reaction, and by which the substrate can be modified into products. So in the enzyme technology, this is concerned with the application of enzyme as tool of industry, agriculture and medicine. Enzymes are the biodegradable, and unlike many inorganic catalysts, causes less damage to the environment. So basically, when a cell grow in the nutrient media, and the substrate is converting into the cell mass, then that is must be a uh, there must be an action of enzyme present. And when the product is formed by this process, those products are called primary metabolite, which is the direct chemical compound produced by the cell mass. And there are another type of products that is produced during this type of reaction, which are less in number. Those are called the secondary metabolites, which are not directly related to the cell growth, but they are very important. That they In the secondary metabolite, we can get some antibiotics and vitamins. Those are all examples of secondary metabolites. So in this enzyme technology, we will study how the primary and secondary metabolites are produced by the action of enzymes using some cell or some microbes in the industry. So let's take a brief look in the structure of enzyme. All are old, but let's take a recap here. The enzymes have two parts. One is non-protein part, and another one is protein part. The protein part is called apoenzyme, and non-protein part, which is firmly attached with the enzyme, called the prosthetic group. There are two types of non-protein part. One is the coenzyme, another is the cofactor. The coenzymes are often they are vitamins, and cofactors are minerals. So all enzymes are protein in nature. There are some exceptions. There are some RNA molecules which have some catalytic activity, but at least much of the enzyme molecules are protein in nature, most of them. So this is the brief description of enzyme, and now I am going to discuss the biotechnological process of enzyme production inside a bioreactor. First, we have to screen. That means we have to choose an appropriate microorganism for the desired enzyme. There are various microorganisms present in our environment which produce various type of enzymes. So we have to first select those microorganisms for the particular enzyme production. Then we modify those microorganisms in some genetic engineering may be required for that. in the laboratory purpose, or after that the modification when it was done, then the uh, we uh, we will go for the laboratory scale production, that is the pilot production inside the lab, and after that the if it is successful, the pilot production, then we will move forward for the mass production of the enzymes using bioreactor for the industry. So take an example, the commercial enzyme production. Here the Aspergillus niger, that is a fungus, is used for the production of pectinase. It is an extracellular enzyme. The pectin extraction was done, and this pectin was used for the production of pectinase enzyme. And it is an pectin is an insoluble substrate found in the cell wall of the plant. It is in the drink industry, that is brewery industry or juice industry. It is extracted from the fruit pulp or fruit peel to appear the cloudy appearance due to the presence of pectin. The pectinase is the enzyme that is used for the industry to break down the pectin. So the effect of pectinase is to clarify the fruit juice and to make it flow more freely. So there are two methods basically for the enzyme production. One is the semi-solid culture, and second one is the submerged culture. The submerged culture and the fermentation equipment is used in the same as the manufacturer of antibiotics, but this production of the the enzyme production using submerged culture needs very much costly, and due to being non cost effective, the submerged culture cannot be chosen for the enzyme production. And this is the semi solid culture where most of the enzymes are produced by this way, where the enzyme producing culture is grown on the surface of a suitable semi-solid substrate. Often it is moist and wheat or rice bran with nutrient. And after that, the media is prepared containing the nutrient salts. The pH is maintained at the neutral level. At least that means the pH must be at 7. After that, the sterilization of the media was done, and the sterilized media is sprayed on the metal trays up to the depth of 1 to 10 centimeters, and the culture is inoculated either in autoclave after cooling or in trays. The high enzyme concentration in the crude fermented material can be obtained using this semi-solid culture. The, there are various enzymes. Those are produced by the semi-solid culture known as alpha amylase, glucomylase, lactase, pectinase, protease, and rennet. These are the some microorganisms. Those are used to produce these enzymes are there. So please go through this slide, which will be mostly effective for your exam purpose. So the advantages of the semi-solid cultures are it involves comparatively low investment. That is, one is the best beneficial for the pilot project, and it allows the use of substrate with high and dry matter content, and hence the yield is higher. The cultivates to cultivate are those molds which cannot grow in the fermenters. They can easily grow with the semi-solid media. They allow this allows the molds to develop into the natural state. So there are also some disadvantages. It requires more space and more labor. That is the main thing which can be need to which must be standardized, I think. This involves the greater risk of infection. That means the contamination chance is higher. It is difficult to introduce an automation in such a system, so the manpower is required in this case. So in previous slides, we studied how the enzyme are can be produced, but what is the purpose of enzymes in the industry? The enzymes are used for the product development. And to develop a product from the substrate, the enzymes must be uh, immobilized on a cell surface, so that the moving substrate on the through the surface of the enzymes, ultimately we can get the product from the substrate. So the enzyme immobilization is a process of confining the enzyme molecules to a solid support over which a substrate is passed to convert to the product. The process whereby the movement of enzymes, cells, organelles, etc., is the space is completely or severely restricted, usually resulting in a water-insoluble form of the enzymes. The immobilized enzyme is one whose movement in the space has been restricted either completely or to a small limited region. So there is the uses of the immobilized enzyme. It can be used for the production of products, important products, and it is it is the economically favorable for the recycling of biocatalyst. The stability is much higher in case of immobilized enzymes if the enzyme is not immobilized, so that so we can further have to recover these enzymes from the product mixture. And so this is very importance of the enzyme immobilization. The methods for enzyme immobilizations are, it can also be bind with some carriers using some physical adsorption, some ionic bonding and covalent bonding can be made for this carrier binding. The enzymes may be cross-linked with the surface, and sometimes enzymes are entrapped into the lattice or microcapsule type or in some nanoparticles inside the bioreactors, so that the enzyme cannot be moved inside the liquid media or liquid substrate where the product production is going on. And this is the methods are described here. I will give this to detail in the notes, so you can go through this notes, and that will be easier to understand the process of enzyme immobilization. So there are some uses of enzymes which can be used in industry. Please go through this slide, and I will give it this as a note of the uses of industrial uses of enzymes. That is, I think it will be helpful for your exam purpose. Thank you.